Welcome students to our practical class. Today the experiment we'll study is number of this experiment is experiment number 2A. This experiment is based on plant tissues. So group of cells having a common origin and performing similar kind of functions are called tissues. And plant tissues are mostly dead so as to provide rigidity and mechanical strength to the plant parts. Plant tissues can be meristematic tissue or permanent tissue, and animal tissues on the other hand are very specialized and living. Most of the plant tissues are dead, they do not grow after a certain aging of a specific plant, but animal tissues are very much specialized and then they show a kind of growth all over the lifetime of a living organism. The aim of our experiment today is identification of parenchyma, collenchyma, sclerenchyma tissues in plants from prepared slides and drawing of their label diagrams. So in laboratory, you will be given permanently mounted slides of parenchyma, collenchyma and sclerenchyma tissue, and you have to observe it under the microscope and you have to write it, the specific observations what you have seen under the microscope in your lab manual. So our first cell type or tissue type is parenchyma tissue. So if you do not stain the specimen and you see the specimen under the bright field microscope, the parenchyma tissue looks like this, and if you give a specific stain to the plant specimen, the parenchyma tissues look like this, a colored one. The identifying features of parenchyma is the parenchyma tissue is the simplest plant tissue existing on this planet. And cells of parenchyma tissue are isodiametric, and parenchyma tissues, the cells which are present in the parenchyma tissue may be oval, round, intercellular spaces are present in between the cells, and parenchyma cells possess large center vacuoles, peripheral cytoplasm with a nucleus. The major functionality of parenchyma tissue is they are used for food storage. They store plant products. It may be waste products also, or maybe certain products which might be used for the plants after a certain time. So the metabolism and the metabolic products can be used and stored in parenchyma tissue. They are the storehouse of a plant body. Modified parenchyma tissues are modified as such to perform photosynthesis, and they provide buoyancy to specific parts of plants. Now we are going to shifting from parenchyma to collenchyma tissue. Now collenchyma tissue, specifically they are present in stem, petiole and leaves of herbaceous dicot plants beneath the epidermis, and they are usually confined to the corners of the cells. It is usually absent in monocot stems and monocot roots. You have to remember that. And the identifying features of collenchyma are the collenchyma cells are somewhat oval or elongated. Each cell of collenchyma tissue possesses large central vacuole and peripheral cytoplasm with prominent nucleus. Thickenings are present at corners of the cells. These are called angular thickenings, and thickening comprise of cellulose and pectin deposition with this specific kind of plant tissue. Intercellular spaces are absent. The cells of collenchyma tissue are densely packed, and collenchyma cells are commonly present below the epidermis in petiole, leaves and stems. Internally, each cell of collenchyma tissue possesses a large central vacuole, peripheral cytoplasm and nucleus, and you have to remember that the angular thickening are only present in collenchyma tissue. So this is the major identifying feature for this kind of tissue. So when you are seeing this collenchyma slide under the microscope, if you can identify the angular thickenings, you can easily say this is the plant tissue which is surely be a collenchyma. The functionality of collenchyma tissue are It provides mechanical support and elasticity to plant tissues, provides tensile strength and rigidity to the plants due to thickening, angular thickening. So the cells are thick enough to give strength and rigidity to the specific plant parts. Chloroplast containing collenchyma cells are responsible for photosynthesis, and being alive, when the collenchyma tissues are alive at a certain age of a plant, it can store food also. And after a certain time, when the growth of the plant restricts or inhibited, collenchyma tissues die. So majorly after a certain time, all the tissues, or majorly the maximum number of cells of collenchyma tissue, are dead. Now we are moving from collenchyma to sclerenchyma tissue. Sclerenchyma tissue are found in xylem and phloem, specifically secondary xylem and phloem, stem, roots, veins of leaves, hard covering of seeds and nuts. Sclerenchyma tissue are divided into two types. One is sclerite and one is fibers. Okay, this figure is taken from a certain botanical book. The reference is given here. And so the sclerite form of sclerenchyma tissue looks like this. This is a schematic diagram. Wherever the fiber form of sclerenchyma tissue looks like this. And you can see that under the microscope, these two kinds of tissues are significantly different from each other. So cells of sclerenchyma tissue are dead with highly thickened walls, and that's why it gives major strength and mechanical support for a specific plant part. The thickenings consist of lignin. It's a biomolecule which is deposited on the cell wall of the cells of sclerenchyma tissue, and that's why the thickening happens. And there are two types of sclerenchyma, what we have discussed earlier. The fiber form, the cells in this fiber form of sclerenchyma tissue are elongated and tapering ends, and sclerite form of sclerenchyma tissue, which are called stone cells, and they are roughly isodiametric cells with narrow cavities. Sclerenchyma cells have pits which act as connections with adjacent cells. Sclerenchyma is present in secondary xylem and secondary phloem. Sclerenchyma tissue is responsible for making the hard covering of seeds, and it provides mechanical strength and stiffness to plant. Now, this 2A experiment of identification of plant tissue, which are parenchyma, collenchyma and sclerenchyma tissue, are discussed here. In the lab, you will be given a permanently mounted slide of this kind of plant tissues. You have to put it under the bright field microscope or the microscope, the student's compound microscope, what we have discussed in earlier classes, and you have to see the tissue with your eye through that microscope, and you have to identify certain features of this tissue to acknowledge the identification of unknown plant tissues. Okay, the writing purpose and portion in your lab manual are already discussed. You have to write all these writing portions in the ruled pages, and the labeled diagrams should be drawn with HB pencil only with proper labeling, and it should be done in the blank or white pages of your lab manual. The whole PDF of this experiment is also provided to you, so please follow the PDF and write down all the details, the identification characteristics and functionality of specific tissues in a proper way what we have discussed earlier. Thank you.